Okay, so this is now your chance to ask some questions. We've got all the speakers back up, but we've also got one extra person on the end, which is Sarah Welsh. Can we pass her a microphone just so she can tell her crucial role in the whole process?
Yeah, so my name is Samantha Welsh. I'm involved with the actual lab side of it, so I'm involved with the applications as they come through. We review the quality of the data that's proposed to be generated, make sure that we're generating good quality data. Then I actually get involved with actual logistics, so getting the samples out of the stores to the researchers. And then we work really closely with the laboratories as well to make sure that throughout the project we're generating good data.
Okay, we've got a question at the back there, please.
I'm David Walker. I'm a member of the Ethics and Governance Council. The question for Aris. In January this year, your president signed an executive order saying, quote, no privacy for non-Americans. In that context, the transfer of any data to the United States of America is potentially the subject of interrogation, investigation by your security services. I'd like your comment on that.
So, I will not endeavor to speak for the U.S. government. I will say that for the studies that we have done, and for this study, the data is de-identified. And so, because we don't have the actual information about who individuals are, even if there was some kind of data breach or if there was some kind of executive order requiring us to give over data, although I don't know that that would meet the current legal standard within the U.S. and there is more than one branch of government in the U.S., but I believe by having de-identified data, the data is still protected.
Yeah, got a question at the back there.
Hi, yeah, it's Jamie Floyd from Genomics PLC. It's a question for Jonathan. We're aware that you didn't release X chromosome imputation for the pilot. We're wondering if you would be doing that for the full 500,000.
Yeah, yes, we are doing it. It's not quite finished, but it's definitely something that we will produce this year. Yeah.
Back there, please.
The presentations have been very interesting in terms of genetic factors in disease, but the other factor that hasn't been covered in this session are the social determinants of life expectancy, healthy life expectancy, and so on. The kind of issues that were covered by a whole range of reports, from the Black report to the Marmot report. Can you suggest ways in which these two factors can be linked together? Because just looking at genetics on its own, while very interesting, it seems to be missing out a large part of the picture. Thank you. That's to anyone who wants to respond. Yeah.
So maybe I'll take that one. I mean, one of the things obviously that I showed with my data was that the socioeconomic may be genetically determined, although clearly it's not actually genetically determined, it's culturally determined on the basis of a phenotype. And so the answer, the way to deal with that is nothing to do with genetics, it's all to do with culture and sociology. I think the kind of thing that we're trying to do is to break down, there actually are real problems in epidemiology where you can have something that you think makes a lot of sense. So for instance, the diabetes genes go with how much you watch television. And so television appears to be the problem. So if you watch more television, you're more likely to get diabetes, and that's additive to the genetic effect. And it's a bigger genetic effect in the people who watch a lot of television. And papers are written like that, and then you kind of say, so genes are going with environment. But the truth is that watching television is predominantly a socioeconomic marker rather than a causal one. So actually making all televisions automatically turn off wouldn't help you. And one of the ways this has been shown in UK Biobank is they found that the best predictor of one of the, as good as watching television or exercise, was wearing sun cream. Sun cream, if you wear that, is protective from getting diabetes. And it goes with the genes for protection from diabetes. And that is because it's a socio-economic marker. And so I think one of the crucial things is that it can start to debunk some of the work that is done, which is missing the fundamental cause. Which I agree with you, there is a fundamental cause of socio-economic which we need to get to. But you may find surprisingly that geneticists are your allies in this, because we're trying to debunk the epidemiology which has tried to give it a different cause in that. So I hope that we can help, even if indirectly. Yeah, at the front here, please. Just while...
Yeah, actually, I don't mind. I was interested in some of the data showing quite small geographical areas that you could infer people came from. How is that done? Is that looking at other data sets? Or?
Yes, there are lots of studies in human genetics, population genetics, where people have gone around the world and tried to collect people who, for example, report that all of their grandparents came from a particular region. And so some of that data is public. And so we can take the genetic data of a UK Biobank participant and compare it to that, and try to apportion various parts of it as similar to those individuals. And so we come up with an estimate of how similar you are to those individuals. And this is an ongoing process. People are collecting more and more genetic data from around the world, and so these estimates will continue to improve. So for example, myself, my ancestry, my father was Italian, my mother was English, but one of her grandparents was Italian, and we didn't really know exactly where he came from. But using this type of approach, we're quite confident now that I'm able to say that my mother's grandfather came from southern Italy, whereas my own father came from northern Italy. And so I think this is the kind of inference that we'll be able to apply to all of the UK Biobank participants, and we will publish the results of that, which will be very interesting.
And is that likely to link to any sort of diseases that we know?
Yes. In all of these studies that we've been discussing, controlling for people's ancestry is very important, because if you don't control for people's ancestry, you can make false discoveries. And so in such a large data set, it's very important that you uncover very fine-scale differences in people's ancestries. And so we expect, once we've uncovered these ancestral differences, we'll feed that back so that all people doing genetic studies will be able to use that. I mean, it has applications to disease studies, but it's also of its own inherent interest. People are naturally interested in where they came from. They have their own, personal history, what they've been told by their families, what they know from their own family records. But the genetic data can be used to augment that.
So the kind of question you might ask is the increased heart rate seen in Scotland, is that entirely environmental, or is there a component of it which is genetic, which, again, it's important to know, even if you can't fix it, for health and what you're trying to do at a population level. Yeah. Rory.
Andrew, one of the things I've found really interesting as an epidemiologist, so not a genetic researcher, is that what's come out of the genotype studies is an understanding of the relevance of genetics to disease that was perhaps counterintuitive, in the sense that the kinds of analysis that Jonathan was showing of the associations of particular parts of the genome with height, or indeed with disease, I think the prejudice was that most of the important information would be within the genes, so within the exomes. But then when these studies have been done of genotyping across the whole genome and looked at associations with characteristics or with disease, actually we found very strong associations in the bits of, a lot of them, in the bits of the genome between the genes, which were often referred to as junk DNA, as not being relevant. So that actually the genotyping studies have suggested that much more of the genome was important beyond the genes, beyond the exome. And I just wondered whether you and the panel might kind of comment about how going from genotype to exome sequence to whole genome sequence, when that becomes feasible on this scale, might produce more and more interestingly different findings.
Maybe I'll just start out with a slight apology about my library that I told you about. And the reason is that if each of the books is a gene, then they're not stacked on shelves next to each other. There is shelves and shelves of nothing, we thought, or rubbish in between them. And really what Jonathan is picking out is actually that stuff that lies between the shelves matters, although it's not in a book. And our feeling is that it must matter because it's regulating the books. It's actually controlling them. And so for that reason, it was a surprise. And I think everything we knew about genetics came from rare disease that was caused by genes. And we've kind of come with that idea that we'd be able to predict disease and that it would all be just one change and we'd find it. And one of the big changes that happened with genetic discovery of this kind of work is this finding that it's outside. So I'll probably turn to some of the groups that are talking about taking it beyond the books and into the rubbish in the shelves between them.
I can say something about that, because one of our biggest challenges is going from associations, which are some change somewhere in the genome, to actually working out which gene that change is actually affecting, because there are all sorts of examples where it's quite close to a gene, it's not affecting the nearest gene, it's affecting something some distance away. So those regulatory mechanisms are really key. And you know, the gold standard would be the whole genome. That would be fantastic. It would be, you know, currently it's a major challenge to analyse that, as you know, and currently it's more expensive, which is why the sequencing project focuses on the exome initially, at least.
There is another good reason to focus on the exome, and I don't want to undersell what you all already have, because you have such rich genotype data and so well imputed. When you marry exome sequencing data, which really, the reason to do exome sequencing data is you get some of the variants that are rarer than the variants that you would be able to impute into. That's really the only difference. So you take the genotyping data, you do an imputation, you get down to, I don't know what your metric is, but usually it's sort of 1% or 0.5% or whatever rarity. And then you add exome sequencing data on top of that. What you're getting are the very rarest variants, down to individual variants. To add whole genome on top of that, all you are really adding, again, are the rarest variants that are in those rubbish regions, if we can sort of term them that way. And so I think you already have an enormous amount of data with the exome data getting rare variants in the regions of the genome that we know have the most impact. And then a broader view across the whole of the genome, but missing only those rarest variants. And absolutely, there are going to be interesting things there, but I consider that high-hanging fruit. And this is low-hanging fruit, and actually one of the things I like to say is it's not even low-hanging fruit. We're the first ones in the orchard. We're just picking up fruit off the ground at this point. So let's do that exercise before we try to climb up the tree.
Take it. Just while we're waiting for the microphone to go, I might just ask a different question, which is one that was raised this morning. So we've got a company like GSK, and one thing would be to say how much money is being put in by GSK in the project doing exome sequencing for 500,000, and really why that's worthwhile, and why, I suppose, to turn the question the other way, why are you prepared to let everybody else have this information after 9 months?
So it's a lot of money. I think the value we get out of it is we want to make genetics fundamental to the way that we discover medicines, which is what I was saying. So we have currently about 10 or 15% of the targets in development in the pharmaceutical industry have got some kind of genetic support. We're trying to up that to more like 60%. And we can talk about why that's not 100%. So the genetics is fundamental to the way that we want to discover medicines. More, you know, why are we not bothered about other people having the data? Well, they'll find interesting things as well. There's no way we can analyse everything that's in that data set, find everything that's there, so there's great value from that. And just as a principle, we're kind of committed to the discovery and that basic science end, doing that in an open way, because we think the synergy that we get from that in terms of we're using UK Biobank as a public resource, as all this other investment into it. It's a fantastic data set. The kind of the deal there is that if we invest in it, then that becomes public as well. I think there's a real synergy there. And I would contrast it with the way that some of the companies have gone about this, which is essentially by setting up, I call them walled gardens, because they're setting up their own studies, they're generating data, and that data is never going to be available to anyone. I don't think that's actually, this is a personal view, not a GSK view, I should say, although it accurately reflects most, the company, I think, which is, I don't think that's an efficient way to proceed, either for that company or for science in general.
Let me just add one thing, because absolutely the discoveries outside of discoveries we make are things that we are going to need and get value from for making drugs. But also, this data is incredibly new. This way of gathering so many individuals and so much phenotype data. And I think one of the things that we're doing is we're trying to be innovative in how we analyze it, but we need other people to also invent new ways of thinking about analyzing this data that we will not invent. So it's not just that we want other people to be able to make discoveries that everyone can benefit from, including us, but we need other people to have other ideas about how to create those insights as well.
I mean, that's clearly right. I think we're a big company, and we might be able to put 10 or 20 people on analysing these data. Jonathan probably has that many people in Oxford. The people in the room added together are going to be many more than that. So.
Yeah, I mean, I think one of the things that I've been struck, as a doctor, I'm a bit cynical of the pharmaceutical industry and their advertising processes and things like that. And I think one of the things is to separate the R&D side of pharmaceutical companies, because one of the things the only thing they need is the truth. You can't make a drug to a reputation. You can't make a drug to a theory. You can only make a drug to a thing that really works. And actually what you start to become aware of, so the reason that he didn't give an amount of money, but it will be approximately on standard costs, it would have cost UK Biobank about £200 million if they were to pay for this amount of extra. It's that kind of region. So that's the kind of money they're putting in. But when you hear that one drug is $1 billion, and this is going to stop them wasting their time. We don't want pharmaceutical companies to waste their time developing drugs to things that aren't real. And that's not for anybody's benefit that that's there. And so actually anything which moves it towards the truth is good. And the other thing is that if we're going to make drugs, if we find a target which is helpful and might be druggable, we need pharmaceutical companies to make those drugs, because that's what they're good at. And the truth is, genetics researchers are never going to become drug manufacturers. So it needs to be a team. But I think the overwhelming thing is truth, which is what we all need if we're going to make medicine go forward. And I think one of the great things about genetic studies is you never get one of these controversies where they say, oh, I'm sorry, we made a mistake, and that data wasn't true. That actually when you get these number of people, when you get this amount of genetic data, the other thing we might hear about is the quality of the data. That the quality is incredibly high in UK Biobank in everything they do, and that means you are getting to the truth. And in the end, what we all need is to understand more if we're going to make drugs or if we're going to try and prevent it with social factors or whatever we do. So I think that's why companies put money in. But I think the bottom line is, in 10 years' time, we'll still be talking about what we're finding in the genetic data. There will be so much to come, and there'll be another chapter. Because all those people who've got diabetes, some of them will then have problems with their kidneys or their eyes, and then we'll understand about that. Some of them will go onto insulin, we'll understand about that. So this is just the beginning. And this data will live on, and the great thing is the genetics doesn't change. It will be there, and we will go on being able to use it for discovery in all the time. So I'm very happy to let anybody have any start they want if they can have the data if we can have the data after that. Sorry,
yeah, just one quick question for the guys from GSK and Regeneron. So sequencing exomes on a population scale is a monumental undertaking. So I can only imagine that's going to throw up a tremendous amount of putative loss of function variants. What's your strategy to change those putative loss of functions into real validated loss of function calls?
So yeah, that is a huge question in the field, which is really when you are identifying a variant that you think has a function, how do you figure out if that function that you think it has is real? This is one of the reasons why I talked a little bit about the virtuous cycle of human genetics and mouse genetics at Regeneron. We are obviously not the only people who do this. This is quite common. But the idea of once you find something that you think truly gives you insight, particularly as it relates to something that you might think could be developed into a drug, you don't just validate the actual variant call, which you should always do before you chase down some apparent signal, but you also try to validate that functional call. You try to make that variant in a mouse, or you make a similar variant in a mouse, or you make a similar variant in a cell line, or you make different forms of the gene within the mouse and the cell line, and you see how they behave. And that process is not it's not computational. It's a little more art than science, right? Biology is exciting to me as a data scientist because it's becoming more computable, but it is not wholly computable, and that is one aspect that I think will always have to be at the bench and in the lab, really sorting out when you see a variant and you think it does something, what it actually really does.
Yes, absolutely. Major hurdle, and what I said about filling in that white space from a genetic association to an end is kind of related to that. A couple of things are really useful. Gene editing and the advances there and the ability to make those genetic changes in cellular systems is a big game changer. The other thing in combination with that that's really valuable often are recall by genotype studies, and I appreciate the difficulties in UK Biobank of that, particularly where one might be looking for very rare variants, which are only in a handful of people, as those loss of function variants maybe. But that's another way of going about it.
Yeah. Could I ask John about the last sentence that he was cut off in his prime and didn't speak about in the context of no study being big enough, no cohort being big enough. And I think what you had on your slide was the routine collection of DNA in healthcare. And I wondered if you'd like to talk a little bit about that. I can see where the advantage is coming from, but some of the obstacles and protections and how you envisage that working.
Yeah, I must admit, I wasn't disappointed to run out of time before we get into that, because it's a big topic. It's happening to some extent on a piecemeal basis already, particularly in the US, where academic medical centres are essentially attempting to consent everybody who comes in to take a DNA sample and then use that data for research use. Clearly, as a researcher, it would be tremendous benefit because you potentially get very big sample sizes. If it's going to happen, all sorts of things have to be lined up. One is appropriate governance, consent processes, and I think benefit to patients as well, because I don't see that happening, and the people in UK Biobank are very public-spirited volunteers, but to really make that happen, I think we're going to have to show that data is useful in medical care as well. So something like Genomics England, the 100,000 Genomes Project, is doing that for rare diseases, and maybe that's the place to start. But I mean, there are probably, I'm a statistician, there are probably other people in the room more qualified than me to speak to this. I'm looking at some of the esteemed epidemiologists at the front, for instance.
Yes.
May I pose what I might call a philosophical question? While I undoubtedly recognise the benefits of what you've been describing for patients in terms of drug development and so on, there is the law of unintended consequences. Now, you're talking about de-identified data, but if this grows internationally, how do I know where my data's going? How do I know what it ultimately will be used for? Because there may be those with less beneficial uses in mind.
So I'm tempted to put that at...
Maybe I should take that as being on the Access Committee. So the crucial question that we ask when the data goes out is, one is this an academic, is it a person who has all the credentials of being a researcher? So that's the first step. And then the key step is the question that they are choosing to ask medically relevant. The issues are, will they do what they say they will do? The crucial thing, and we might ask Sam about this, is that my understanding is that there is no way that any data that would allow it to be identified is ever gone, is never left, is never available to anybody. So it cannot be shared. So what can't be shared is any information about you. Is that right, Sam?
Yeah, that's right. So in terms of the lab side of things, when we send samples out, we send a list of IDs. So to us, all participants are known by a 9-digit identifier. We don't know your name, address, anything like that.
The issue is the use which is put ultimately.
So could you give a use that you would be concerned about, which would, the kind of thing that you wouldn't want it to be? That would be helpful for us to be able to answer that.
I mean, this is in the realms of fantasy at the moment, but suppose you find that a third of the population is susceptible to a certain kind of, let's say, fertilizer or whatever. Now, the good thing is that it could help 2/3, but it wouldn't help 1/3. But someone with less good intentions might think, what could I do with that country that we're not very happy about? I was taking it beyond the UK Biobank to the fact that you're talking about federated information and sharing, and ultimately, science is international.
Yeah, and I think probably if we're being realistic, we can't give you a reassurance that science will only be used for good, because history tells us that's not the case. So obviously it's outside UK Biobank, because if they wanted to do that, if they wanted to find if it was a third of the population, they could do that with 100 samples from their backyard. You know, they wouldn't need this number of people. So it can't be regulated at UK Biobank level. So it will be interesting. There will be variants of the gene that alter how you metabolize drugs, and they may also metabolize poisons as well. And I think it's one of those things where it's up to everybody in society, and why have we not had a repeat of atomic warfare? You know, those kind of questions, which is all about science and it being used to bad use, even if it was. And so I think it's probably outside UK Biobank, the use of these things. But I don't think we can regulate as a scientist. If you ask me, can I stop the particular scenario you're talking about, I don't think I can, unless I tried to remove all DNA from everybody. But the benefits, so it's again against the question of benefit versus harm. And I think the benefit that will come from new drugs, new understanding for health, that's happening already against a theoretical chance that it could be used. So therefore, I think we have to continue with genetic research, but always be aware, and it will be whistleblowers, it will be people on the inside that have in genetics, who if there's anything that could be used, they will be the people that understand the most. Those are the people, it's us as geneticists who have to always think, could this be put to bad use? and how would we stop that when a particular situation comes up? Does anyone else have any answers to? Rory, yeah, please.
In a way, I think there are two aspects to the question. So there's one, relates to UK Biobank more narrowly, if you like, about how do we ensure, as far as we can, that the UK Biobank data are used by researchers in ways that help improve health, which is our aim. And how do we ensure that they are doing that? And that was really what Andrew was addressing, I think, around the access policy, about ensuring that people are bona fide researchers, that they have a particular piece of work they're going to do, that they report what they have done. So, for example, in the agreement with GSK and Regeneron, they have to put into the public domain their findings, either publish them in journals or provide annual reports to us about what they found, or there may be things that they patent, as academic researchers can do as well, that will again get into the public domain. So we know what they have found. Of course, one might say, well, but have they told us everything that they've found? And I think the answer to that is that it's an open resource. So if they've found something that's significant and haven't told us, then those data will become available to other researchers who will then find it. So we have a kind of process which is, by its nature, transparent. So they found good things or bad things. And I think that goes to your broader question about finding things. If you know something and no one else knows it, it's a weapon. If you find something and you don't protect it in some way, and then someone else finds it, you've thrown away your weapon. You've lost your protection. So actually the incentive on people, if they find something significant, is to make it public, to make it clear they found it, to patent it if they want to patent it, so they protect it. If they hide it and don't tell anybody, then someone else will find it and they will get the glory, or they will get the patents or whatever. Because it's this open and transparent process. So in some ways, this provides a protection against the more general concern that you express around having information that could be abused. That can be abused if you're the only one who knows it, but if other people can find it out and know it, then they have a defense against the weapon, and you've essentially thrown away your advantage.
Okay, so just one final question, in a way picking up what Rory said, because I think for people working in this area, one of the exciting things is that this is available to everyone. That there isn't a researcher in the UK who's bona fide at a proper institution who can't have it. And I just thought we might ask Jonathan to say how he thinks this has altered research, because it used to be very structured. There were leaders in genetics and they would have teams, and it was very structured, but things now are such that anybody, even if they're a junior member of the team, can come up with new research. And have you seen any examples of things changing because of the availability of the data to everyone?
Yes, I mean, collaboration in our field, I think, has changed dramatically over the last 10 years. The availability of many people to data sets that traditionally might have just been kept within an individual lab. That model has broken, has broken substantially. People have realized that they have to work together to collaborate, and that's, you see that at all levels. The senior people in the field need to work together, build collaborations, and then that trickles down. You get junior people in the field meeting each other, starting collaborations, and so I think it's extremely beneficial. And with a resource like Biobank, where lots of different groups working on it, and all feeding their information back to each other through papers and conferences, I think it's a very good thing.
Excellent.
Just one quick comment.
Sorry, yeah.
I think this will be the single most important data set in genetics, full stop, period. This is going to transform the way people do medicine. This is going to transform the way people do genetics. It is incredibly valuable to the field. So people should be incredibly proud of that.
Yeah, I think on that point, I would heartily agree with that. Biobank is amazing and it is available. Many people are talking about massive studies and talking about making it freely available. Biobank is doing it now, is doing it quickly, and is doing it at a very high standard. It would all have been impossible without the 500,000 people who took part. And so from the genetics fraternity, just a massive thank you to everybody who's made it happen. Thank you.